Okay, I think you can share your screen now. Okay, sorry about that. And um, okay, so hello and greetings everyone. This is the 10th of our Afro-Longevity Movement educational program. In our attempt to ease the burden of one person doing everything, we resolved that the problem before us as the members of the Afro-Longevity community is a lack of people on ground. And because Africa is so big, we wanted to raise a longevist or activist or enthusiast across the African continent so that we can help translate and take the message across to the grassroots level. All right, so we've been doing this program in an attempt to ensure that the message of longevity spread across and that institutions start finding meaning and interest in the longevity, advocacy, leadership, and everything that has to do with improving the quality of life of the people, including um, talking about policies that help nations to bridge the longevity divide. And one of the finest people in the longevity community is Dr. David Mayer. He has been a very supportive member of what we do in Afro Longevity. I would say that the desire that it seems that he is even more serious than the people here because he is trying to help us with a whole lot of ideas. Uh, and, in ensuring that we are very successful in what we want to do and how we want to do what we do. And his proposed topic as a faculty is something that is very important to us, especially our research and development department led by Okeke Fani, with regards to our interest to have aging clocks, and we are building some systems, AI tools to ensure that we do something different in this part of the world. So I would like to say a little bit about Dr. Mayer before yielding the mic to him to tell us a little bit about his background and why this is very important to us today. So he is an independent PI at the Institute of Genome Stability in Aging and Diseases at the CECAD Research Center of the University of Cologne since 2025. So he's done with his master's and PhD in bioinformatics in Germany and PhD School of Aging Research at the University of Cologne, Germany in 2024, and conducted his postdoctoral research as the bioinformatics led spearheading the data integration project of the research group on physiological causes and consequences of genome instability. So he's a man of too many hats. So I will yield the mic now to him, allowing him to tell us a little bit about the background and what he thinks Africa should do better, and then the teachings. Thank you so much, Dr. Mayer.
Yeah, thank you, Ozzy, and thank you for the really kind introduction. I'm really happy to be here, and I think it's really amazing what you're developing here, and happy to also be part of this in the future. So yes, as was already mentioned, I'm David, currently PI in Cologne, and I would like to talk to you today about the development and application of aging clocks. Should be probably around 45 minutes or so, the the presentation. So to start off with, you know that everyone of us ages, and this can really happen at different speeds. Some of us age faster or slower than others. The question is why? But to be able to understand why, we first need to be able to measure the difference in the biological age. And for this, we first need to understand what the difference is between the biological and the chronological age. And I want to give you a brief history introduction about this topic. Roughly 70 years ago, Benjamin published this article where he argued that one man may be 60 years old, while another may be 60 years young. So the question is really, how old is old? And the calendar really does not hold the key to this question. So one 60-year-old might be close to death and multimorbid, while another may still be running marathons. So what is this difference? How can we measure it? And 70 years ago, Benjamin already proposed a way to measure it with a simple questionnaire. And here, this is, of course, something that you probably have seen before in your medical doctor's office. It's just questions about your diet, your eating habits, smoking, sleeping, and so on. And you get, in the end, some kind of score, which then tells you whether you're biologically young or old. This is, of course, highly subjective and doesn't really tell you a lot about the underlying causes of aging. So it was a good start, but we needed to go something deeper. Oops, sorry. So, roughly 15 years later, this topic was brought up again by a Japanese group that was interested in age-related changes in Hiroshima subjects. Why were they interested? It was in their 40s and 50s that researchers figured out that radioactive irradiation does not only lead to cancer, but seems to increase the aging rate of model organisms like mice as well. So these mice seem to age faster. And these researchers here wanted to know whether the same is happening in humans as well. And 15 years before this publication came out, there was, of course, the atomic bombing of Hiroshima and Nagasaki. So many people that underwent a lot of radioactive irradiation. And now they wanted to know, were these people aging faster than a control cohort? And during that time, we, of course, didn't have aging clocks yet. So initially, the first results were based on human estimates. So in this case, it was really a nurse's estimate. They asked nurses to estimate the age of the people, the chronological age, and use this as their readout. And first of all, you can see here on the right that these nurses are really highly accurate in predicting the chronological age. So they are less than 1 year off, much better than I could be. Males were generally a bit younger predicted and females a bit older. But the more interesting part is here in the different groups. Group 1 were those individuals that 15 years earlier had high irradiation. They were close by to the atomic bomb. Whereas Group 4 had no radiation. They were living in a different city. And the idea was now, of course, that Group 1, because they had more irradiation, would be predicted older, and then it would decrease gradually. But as you can see, there's no clear pattern in the data, so the results were negative. Based on nurse estimates, there does not seem to be any trend in the data. There's no biological age measurable. Now, this is, of course, still subjective, and these researchers now thought, okay, maybe we can do it somewhat differently, and they went to biomarkers of aging. What is a biomarker? A biomarker could be anything that you measure, from measurements of blood, your skin, hair, blood pressure, and so on. And you will always find some biomarkers that tend to go up with age and some that tend to go down with age. But every single biomarker is really not that informative. So instead, over the recent years and decades, machine learning models have been developed, often really something as simple as a linear regression, that then give rise to predictors of the chronological age and now subsequently also biological age. And this Japanese group did exactly this. They used some biomarkers, which you can see here. So from hair graying, skin elasticity, blood pressure, and so on. And they built the first real aging clock with just a linear regression. So something that you often do even in school. And you can see here on the right that this first clock was already quite accurate. You can see the actual age on the x-axis and the predicted age here on the y-axis. And for males and females, it seems to be decently accurately predicting the chronological age. However, and for this they did not have a figure or a table in the paper, they also tested again the 4 groups, high irradiation to no irradiation. And again, they did not see any differences. So also with this biomarker-based clock, there was no difference in the biological age of the irradiated humans. So again, a negative result, and this put a bit of a stop to the whole field. And it took quite a while until it got taken up again by researchers. In the meanwhile, in the 70s, 80s, there were several discussions, several papers that discussed that aging clocks might not be feasible. Maybe they are not doable. Maybe every single cell in your body has a different aging clock. And maybe even if you have a clock, it doesn't tell you anything useful. So it was really not really going forward. And this only changed then when epigenetics became more accessible. So just a brief explainer what epigenetics is. This is a pretty broad term. You have on the DNA small modifications, DNA methylations that regulate whether a gene is active or not. This is the most important for us today. But then, of course, epigenetics is more than that. So the DNA is wrapped around the histones, and the histones have a lot of different modifications that are a really complex regulatory pattern, where you have acetylations, methylations, and all other kinds of modifications that interact with each other, which makes it really difficult. The DNA methylation, however, is what we want to focus on today. And here it was already known by the time when the first aging clocks came out that every single cell has essentially its own epigenetic makeup. So the genome is, of course, the same. You have the same genes in every cell, but a skin cell has a different task than a liver cell. So you need different genes or proteins to be active. And this is essentially regulated by DNA methylation. So if DNA methylation is there, usually the gene is repressed. If it's not there, the gene is active. So every cell type has a different DNA methylation makeup. And now, why is this interesting? This came from a study from a different field, actually, where they were interested in twins. So this was a twin study that more or less kickstarted then the field again. In this study, they were interested in monozygotic twins and why they tend to diverge with age. So monozygotic twins, of course, are genetically identical, and also on the epigenetic level, they are essentially the same at birth. But with time, the older these twins get, the more they diverge, of course, so the more different they look. And the question is, how could that be if everything is the same at the beginning? And here in this paper, they saw that it's really the epigenetic differences that tends to increase with lifetime. So this DNA methylation is changing within them. And this is, of course, interesting for the aging field when they saw this, because here now we have a biomarker that does change with age. So naturally, people wanted to test whether we can build an aging clock. And exactly this has now been tested then by Bocklet in this first epigenetic aging clock paper a decade ago. Here they used just a small data set, but they saw that they can build a predictor of the chronological age just based on the epigenetic changes over time. This was interesting, but there was still a lot of skepticism in the field because, as I've just told you, every cell type has a different epigenetic makeup. So the question is, if I have an aging clock that is built on skin cells, does it tell me anything beyond skin cells? Because the liver has a different DNA methylation setup. And it was then only until Steve Horvath in his landmark publication a few years later showed that indeed you can build an aging clock for essentially all cell types in the human body. So irrespective of the cell type, you could get an aging clock working that predicts the chronological age to high accuracy. And this really surprised me in the field because the thought was that because every cell type has a different DNA methylation setup, there shouldn't be this conserved pattern in the data. But this is exactly what you found, that there are patterns in the data that are irrespective of the cell type changing with age. Now, this is based on chronological age. And we, of course, don't want a perfect chronological age predictor. If you would have a perfect chronological age predictor, it would not tell us anything about biology. We could just look at the passport, essentially. This, of course, would still be interesting for forensics or the police department because they could figure out the age of an individual. But this is not what we are as interested in the field of aging and longevity. And here the hypothesis comes in that the prediction error contains biological information. So if I'm 50 years old, but I'm predicted 60 years, then I'm 10 years older biologically, so I'm fast aging. And the theory is then, or the hypothesis is, that I'm aging faster and probably also die earlier. And this is exactly what then has been seen. Aging clock predictions are associated with mortality indeed. So I just show you here one example. This is not based on Horvath's clock, but on phenoage, which is a later generation. But here they saw that those individuals that are predicted younger, here in blue, so 10 years younger than the chronological age, they tend to live much longer compared to those individuals that are predicted 10 years older than their chronological age. These tend to die much earlier. So here on the x-axis, you see the chronological age, and on the y-axis, the patient survival. And you can see that these are clearly different. Those that are predicted older indeed die earlier. So there is value in these clocks. Applying them definitely does seem to show you an association with age-related diseases, but also mortality. However, there are still several problems in the field. This was is from a nice publication from 2 years ago, validation of biomarkers of aging. And one problem is that almost all aging clocks are built on cross-sectional data. With cross-sectional data, it means we have one large cohort, let's say 1,000 people from 20 to 80 years, but every individual is only sampled once. So we only have one blood draw, one measurement from one individual. So we don't have longitudinal data. But in theory, of course, every individual should have its own aging trajectory. So we cannot know this without the actual longitudinal data. So cross-sectional data does not allow proper validation and identification of causal effects. And on top of that, almost all data that is used currently is from Caucasian populations. So we lack a lot of diversity. There's almost nothing from Asian or African populations, for example. There has been shown that if I train an aging clock, for example, on a Caucasian population, it often works not as well on a different population. So there's definitely need for more diverse data sets. First of all, for the populations that are not represented at the moment, and then also to get a more universal understanding of what aging actually is. And for this, we need then essentially longitudinal data from a diverse population where we sample the same individuals multiple times over years, ideally, of course, and then can really have a predictive value which can really be validated easily. But the data is just currently not there, and this, of course, takes a lot of time and effort to actually generate such a database. A further problem is that aging clocks, specifically also epigenetic clocks, which are currently still the cornerstone, are highly cell type composition confounded. What do I mean by that? Just one example of cell type composition changes in blood with age. Here I show you from this publication how different cell types change with age. So on the x-axis here is always chronological age, and on the y-axis is the cell type fraction of a certain cell type. So for example, here in this case, this is CD8 T cells, naive T cells, and you can see that over time, this cell type decreases dramatically. So it's rather abundant in young individuals, but it then decreases dramatically with age and almost reaches zero in certain individuals. On the other hand, for example, monocytes, they tend to increase with age. Really showing that with age, we have a big cell type composition change. Problem is that all these data sets that we generate are usually a mixture of cells. So usually we have a blood draw, and then we just mash up everything that is in there and we sequence it. But now we don't know, is the change that we are seeing because the cell types change? So I just have less CD8 T cells, or is it that the cells itself change? And this is, with the current data, for most of the data sets, not really easily doable. And why is this not relevant? Because this leads to such effects that stress-induced cell type composition really changes and confounds the predictions. Here, just two recent examples. On the left side, I showed you data from an emergency hip surgery with 3 different time points. Time point 1 is directly before the hip surgery. Time point 2 is directly after the surgery, where the patient is inflamed and under stress. And then time point 3 is after a while, where the patient is healthy again. And you can see that directly after the surgery, the age of most individuals jumped up to 10 years, showing that stress and inflammation really drives up your predicted age. And this is even more dramatic if you look on the right side. In this case, this is a study that looked at viral infections. Here in this case, you can see the healthy cohorts on the top, for example. This black line is the 0 line. So those individuals are roughly predicted their chronological age. And then you can see here, once the viral infection hits, in the acute phase, these individuals are up to 25 years older predicted than the chronological age. And this then reduces slowly again until these people are healthy, and then they are back to the baseline. But this is, of course, not what we ideally want to call biological age, something that fluctuates as easily with just viral infection. This just measures essentially your inflammation setup. This is not bad per se, but it gives you a different information. So as I've shown you, these aging clocks still are associated with mortality, for example, and age-associated diseases. But the way they measure this is not really the biological age, but just inflammation. We know, of course, that inflammation increases with age, and this is a useful readout, but one has to be careful on how to interpret this. And this becomes especially visible if we look at the circadian rhythm. So even the circadian rhythm impacts the predictions. The circadian rhythm is, of course, changing over the day. It gives you the wake and sleep cycle. And in this publication from 2 years ago, they showed this really nicely. They sampled the same individual over 24 hours. So really at several time points, they had the blood drawn, I think. And then they measured an aging clock. And you can see that the aging clock predictions really follow the circadian rhythm. So it varies for the same individual from 55 to 62 years. So almost a 10-year difference, although this is the same person at the same day getting measured. And why is this so? Because also during the day, your cell type composition changes. In the morning, when you wake up, you have a spike in stress hormones, your cortisol, to make you ready for the day. You have a different cell type composition. And then once in the night, if you relax again, your stress hormones, of course, also reduce, and your cell type composition changes in the blood. You have different immune cells in the morning, in the midnight, or in the night. And this is exactly what's getting measured here. And this is, of course, not ideally what should be happening, right? You don't want that on the same day for the same individuals, you have 10 years of a difference in the prediction. This is not useful. And some remaining problems is that DNA methylation is generally hard to interpret. So with DNA methylation clocks, you get your predicted age, but that's about it. You don't really know what to do with it. There's no easy way to interpret it. You don't know what you can do against it, or whether there's anything else in your data. It's really hard to interpret. Then predictions are really not easy to validate in humans, especially. We just don't have enough data. And we have, of course, highly variable environment in genetics. And as I mentioned before, different populations really behave differently here. And really to validate this, ideally what we would need to do is to measure individuals and then follow them until they die to see whether there's a real good association. And this is, of course, difficult. And as I mentioned, clocks are confounded by cell type composition changes. This is also why a few years ago, we have set out and built our own aging clock. So this is now roughly 5 years ago, not on DNA methylation, but on transcriptomic data. And here we try to also do something about this current problems. So first of all, we did not use DNA methylation. We used gene expression data, which is generally easier to understand and generate hypotheses. This is not in humans, but in C. elegans, so nematodes. It's a model organism that just lives 2 to 3 weeks. But here we can really easily then test and validate our hypothesis. And in C. elegans, we almost have no cell type composition changes. So it's much easier to interpret than later on. I'm skipping a lot of details here. Transcriptomic data is generally harder to work with for aging clocks because the data is noisier. You can have a look at the paper, or we can also discuss it later. But what we saw is that we can build a highly accurate predictor. And now because we have gene expression data, we can directly have a look and see what genes and pathways are actually important for the age prediction. And here, our clock, we call it bit age, finds several systemic pathways to be highly enriched and important. We have, for example, signal peptides, we have the innate immune response, neuropeptides, and a lot of channel and transporters that seem to do something with aging. So there's something going on or maybe wrong with aging, in this case in C. elegans. And now, because this is in C. elegans, we could, of course, now say, okay, the innate immune response seems to play a role. What happens now if we knock it out or overexpress it? What happens to these worms? Do they age faster or slower, for example? So it's a nice model organism. But I don't want to go into this direction. I rather want to talk about another still problem, also with gene expression data here. This came from a publication a few years ago, where they saw that many aging clocks can be found. So the question is really, how can we interpret them? So this paper focused on DNA methylation, but the same is true also for gene expression data. So here we found, with gene expression, this aging clock, we found these pathways to be important. But what now this paper showed is that you can build other clocks with other genes. So essentially, for example, let's say we have 10,000 genes, and we find 500 genes important for an aging clock, for example, this year. Now, if we remove these 500 genes, we are left with 9,500 genes, and now we train again an aging clock on the same data, just with less genes. Then we will find an aging clock that is almost exactly as accurate as the first one, but has different genes and also different pathways. The question is, how can we even interpret it then? So what is the underlying signal of aging clocks, and how can the prediction be explained? And this is also motivated by another fact that we know that DNA damage, for example, drives the aging process. If we have more DNA damage, we can accelerate the aging process. If we have better DNA repair, we live longer. This is also true across species. But DNA damage is a random process. It's stochastic. It happens randomly in the genome. So the question is, how can a random event lead to something that is predictable? And this is something we wanted to explain in this publication 2 years ago. We wanted to see how much a stochastic process alone could explain. So do we need a program, or is a stochastic, a random process like DNA damage sufficient? At the moment, it sounds like if we have a random process, I shouldn't be able to produce something that is reproducible and almost deterministically able to predict something. And this is something we wanted to see here. And here I want to walk you briefly through our process. We started with a completely artificial setup. So in the beginning, we did not have any biological data in here. We just wanted to see whether it's possible in theory. So we start with a ground state. It's just a starting point. In this case, nothing biological. It's just random values between 0 and 1. And then what we do is we simulate a stochastic process. This can be visualized by such a golden board. You probably have seen it before. You throw some sand or small balls in it, and then in the end, you get this nice, normal distributed curve. And we did exactly this. We simulated small random numbers with such a normal distribution, set that on 0, so it's equally likely to be negative or positive. And we just added these values to the ground state independently. And doing this once, we just defined to be one h step. So the ground state is h0, and adding this noise one time means h1. And now we can, of course, do this multiple times. So not only once, but let's say 100 times. We always start from the same ground state, but we add noise several times. So from 1 time to 100 times. Now h1 is, of course, still highly similar to the ground state because we just added noise, small noise 1 time. Adding noise 100 times, of course, in the end looks completely different. So this data will look completely different from the ground state, completely noisy in the end. And now we wanted to see whether we can use this data to build something similar to an aging clock. So we use this data for a regression model. So it's an elastic edge regression. It's a standard linear regression that tries then to predict the simulated age. So we try to predict how often we added noise to the data. And then we, of course, need to test it on something. So we generate some additional data from the same ground state independently. And now we want to see whether we can get any prediction at all. And at the beginning, we saw exactly nothing. There's no prediction possible at all. So if we just accumulate stochastic variation, if we just add more and more noise, there's no pattern in the data. Noise, by definition, of course, does not have a pattern. So we don't see any prediction. So the simulated age, how often we added noise, cannot be predicted. It lies completely flat. But then we thought this is also not how biology behaves. In biology, we usually have some kind of constraints. For example, DNA methylation. In a single cell, it is 0 or 1. It's either there or it's not there. And if you now have a bulk sequencing, so usually the data that you look at, it is between 0% and 100% methylated. You cannot be 110% methylated or minus 10% methylated. That doesn't make any sense. And now just by restraining our data to be within some limits, we do see that we suddenly can build highly accurate predictor of the simulated age. So it's exactly the same. We just add noise. We just say the data cannot go below 0 or above 1, for example. Again, this is completely artificial data. There's no biology here in it all. But we do see that in theory, we can build predictors, even if it's completely noise that we use. And this now made us think, of course, how would age predictors, aging clocks, behave on data with accumulating stochastic variation? So if I have an aging clock like Horvath's DNA methylation clock, and I use it on a young human, then of course the predicted age is highly accurate. It should be, if the human is 20 years old, roughly 20 years. And if I now use the data and just add noise to it, how would the predictions look like? So if I have the same setup as before, I just start with the biological time point, and I use now an epigenetic clock. What would it look like? And to our surprise, epigenetic clock predictions correlate linearly with the amount of stochastic variation in the data. So again, here on the x-axis is the simulated age, so how often we added noise to the young human sample. And on the y-axis is now the public clock, the Horvath epigenetic aging clock. And you can see the more noise we add artificially, the older the sample gets predicted. And this is not only true for Horvath's clock, but essentially all aging clocks that we tested. The They all are linearly correlated with the amount of noise that we artificially add to the data. And now, of course, this made us wonder again, can we even do the reverse? So can we use a simulated data set and use it to build a predictor of age? So same as before, we have training data. We use added noise. We train a predictor that tries to predict the simulated age. But now the test data is actual biological data. So it's human blood samples. How would that look like? And to our surprise again, this worked surprisingly well. The stochastic clock can predict the chronological age of biological samples. So here on the x-axis is the chronological age. Every dot is a human blood sample, DNA methylation. On the y-axis is the predicted age with our clock. And just to rephrase that again, this clock was just trained on one single biological sample. The starting point was biological, a young human blood sample. Everything else was just noise that we added artificially. And this clock can still predict such a linear correlation here. So this is chronological age on just humans. What about other species? And for this, we had a look at the pan-mammalian aging clock. So this was a nice publication from Steve Horvath and colleagues 3 years ago, where they generated DNA methylation from all these different mammalian species. So you can see here they have mouse, they have humans, whales, elephants, dolphins, zebras, and so on. And they built an aging clock that can predict all of these species almost equally well. So how they did this is they have here more than 10,000 DNA methylation samples. They take one species out. Let's say they take the elephant out. They train on all the other species. And then they predict the elephant. And you can see here in this line around the circle, the accuracy. It's a bit hard to read maybe, but this is the correlation value here. And you can see it's above 0.8. So almost 0.9 for many of these species. It's highly accurately predicting the age of these individual species. And now we thought, can we do the same with a stochastic clock? So we used, in this case, one single sample from the bottlenose dolphin. We just added noise to it. We trained it the same as before, and then we used all these more than 10,000 samples to see whether the prediction would work. And surprisingly, for most of the species, it again works remarkably well, with correlations above 0.8 for many of them, showing that one single biological sample is sufficient even for a pan-mammalian aging clock. So the question then is, this stochastic noise, what we did here is completely artificial. We just added it in the computer. In biology, where does it actually come from? What are the drivers of stochastic variation accumulation? It must be happening also in biology if this is happening. And this is something we published a perspective on last year. So have a look if you're interested in a more broad discussion without much new data. And just briefly describing what we explained there is that biological systems are inherently stochastic. So what do you mean by that? Within a cell, every protein, every enzyme is moving randomly. It's called Brownian motion. So it's just moving randomly in your cell. Then we have, as I mentioned before, stochastic DNA damage. And this can, of course, be repaired without anything left. But you have also sometimes stochastic somatic mutations happening, or sometimes the damage is just not getting repaired and it's just incomplete and it just stays in your body. Then we have also stochastic epimutations. So the DNA methylation can also mutate. This happens, for example, during cell division, where you have to copy everything perfectly, not on your DNA, but also the DNA methylation. And sometimes it just not gets copied perfectly. Nothing is 100% accurate. Transcription is inherently noisy and stochastic. We have even two different types of noise. We have the extrinsic noise and the intrinsic noise. Extrinsic noise is everything that comes from the outside. It can be the environment, influences, hormones, temperature, and so on. And the intrinsic noise is really how transcription works. It behaves in bursts, and depending on how the polymerase sits on the DNA. Translation is, of course, also a stochastic process. The ribosomes have to bind to the RNA. We have a stoichiometric balance that needs to be kept. The ribosome abundance plays a role, the tRNA abundance, and then once everything is translated, of course, also protein folding is a stochastic process. There can be many mistakes and errors happening. And in the end, this leads then to a gradual decline in network stability. So in the young and healthy individual, you have like these connections between genes. So gene A might influence gene B, which might influence gene D, for example. But with age, due to all these different stochastic mistakes that are happening, this network degrades with time. So maybe suddenly gene A is more strongly activating or inhibiting gene C, or the edge between D and E is completely missing, and suddenly it cannot cope with certain stimuli anymore. And what we think is that stochasticity is hierarchical. So DNA damage, once it appears, it has to be repaired. And DNA damage induces or can induce hemimethylation. So if you have here, for example, a single strand break or some other event happening, then the DNA has to be repaired, but also the DNA methylation has to be copied again. But during DNA repair, mistakes can happen, and suddenly this is not methylated anymore, but unmethylated. And this, of course, then can propagate. Or it could happen that DNMT1, this is the methyltransferase, mistakes a mismatch base as the methylated C. So this can happen during replication, where initially you have a mismatch here. This is maybe faithfully repaired afterwards, but DNMT1 sees it already and suddenly methylates it, although it's not supposed to methylate. And once then replication is ongoing, you suddenly have a methylated base where you don't have, where there's not supposed to be any. And of course, if you have a somatic mutation, you can have an irreversible loss of DNA methylation. So here was supposed to be a methylated cytosine, but due to a mutation, suddenly it's completely gone. And then, of course, it cannot function anymore. And now if you have a repressed gene, so if the promoter is methylated, it's usually repressed. Over time, with stochastic variation, the only way this can go, it's becoming less methylated. Because it's 100% methylated, it can only be less methylated with time. With every single mistake during cell division, during DNA repair, you will have some mistakes happening, and on average, you will have less and less methylation. And at some point, suddenly, you will have not sufficient DNA methylation, and the gene that is supposed to be repressed is suddenly getting activated again. So stochasticity mistakes on the DNA methylation level can then trickle down to the transcription level, and then, of course, also further to the protein level. So the conclusion so far is that the chronological age can almost perfectly be predicted. The caveat is that this really then leads to a loss of biological information. We want some variability in the data. Aging clocks are confounded by several things, especially cell type composition changes, and stress and time of the day really impact predictions, which is not what we want. There have now been attempts to resolve this. It's getting better, but it's still a difficult problem. And what we think the real underlying explanation of aging clocks is increasing stochasticity. And this is most probably due to this imperfect maintenance system. No copying mechanism, no repair mechanism is perfect. And even if it's 99.99% accurate, there will be mistakes happening. And as I've shown you here, if you're 100% methylated, the only way mistakes can go is in the other direction. So with time, you lose repression. And this is exactly what we do see. So I've told you a lot about how we can develop these aging clocks. But the question now is, how can we actually use them to hopefully accelerate aging research? And here there was this nice publication a few years ago about the biomarkers of aging for the identification and evaluation of longevity interventions. It's a really nice read. And here they divided this into 3 different topics. One is predictive or prognostic. This is how most people use it nowadays. So you want to predict a patient, for example, and get some data out of that. So for example, again, if the patient is 50 and he's predicted or she's predicted 60, then, oh, sorry, yeah, there's a question?
No, it's not a question. Sorry, continue.
Okay. Okay, otherwise, let me know. So if the patient is 50 and he or she's predicted 60, then this is an indication that this patient might die earlier, that he or she might develop some diseases, and that maybe the medical doctor should do some additional checkups on the patient. Then a second part is response, they called it. And here the most important is surrogate endpoints. If this works, that would be really revolutionary for the field. And what they mean with this is, if you have a clinical study where you want to see whether treatment medication has an impact on aging, you would, in theory, of course, have to wait until the individuals die to actually see whether it has an impact, which takes decades. We cannot wait for that. And the hope is that with aging clocks, we can speed this up, so we can, after the intervention ends, just measure the aging clock and see whether the predicted age is reduced. And then if we see a 10-year reduction in the predicted age, hopefully this also means that these individuals will die 10 years later. However, due to all these problems that we're still facing, currently we are not at this level yet. We hope to get there, but we are not there yet. It's still good to try these as surrogate endpoints in clinical studies, but we still have to do lifespan curves at the moment, sadly. And the last part, which I want to briefly focus on, is discovery. And here, this is something that we published just recently that I briefly want to explain. Here we ask the question, can aging clocks uncover neuron-specific aging patterns to guide neuroprotective drug discovery? This seems quite specific, but this question is motivated by the fact that, for example, in Alzheimer's disease or Parkinson's disease, there are usually specific neuron types that degenerate earlier than others. And the question is, why is this happening? And currently, we don't really know whether every single cell type and organ in your body ages at the same rate, or whether there might be some cells that age faster than others. And this is really where aging clocks come in useful. And here we ask this question for neurons, and we made use not of human data initially, but of, again, the C. elegans nematode data with our previously established bit age aging clock. And here we saw a really interesting pattern. So why C. elegans? C. elegans has the benefit that if you have 2 worms on the plate, these are identical clones. They have the same genetic makeup, they develop the same, they have exactly the same number of cells, and the cells do exactly the same, essentially. C. elegans has roughly 300 neurons, 128 neuron types, and they always do the same thing. You have certain motor neurons, you have sensory neurons, some that sense heat, some that sense salt, for example. And we know exactly where they are in the worm, and we can also track them in live worms. So we can mark them with a fluorescent marker and track them over the aging trajectory. What we don't know is whether they age at different rates. So we used a public data set where they sequenced all 128 neurons at a young chronological age. So these are all from the young animal. And we now applied our aging clock to see whether all of the neurons age exactly the same or whether there are differences. And interestingly, and you can see this here, is that there are indeed quite big differences. The youngest neuron, so it doesn't matter how these are named, is predicted 98 hours, whereas the oldest neuron in these young animals is predicted 177 hours. So it's almost a 2-fold difference in the predicted age. Now, it could, of course, be that this is just an artifact, that the clock didn't work, so we needed to test it. So we chose 3 young neurons and 3 old neurons and checked them in living animals. And you can directly see this. We saw that the predicted age is a robust predictor of degeneration. So here on the left side, I'll show you the young neurons, predicted young neurons. On the right side, the predicted old neurons. We followed the worms from day 1 to day 7 of adulthood. So day 7 is mid-age. And color-coded, you see either healthy neurons in blue, in orange, mildly damaged neurons, or in red, severely damaged neurons. And you can directly see on the left side, you have more blue. So there are more healthy neurons. And what is surprising is that those neurons that are predicted to be old, they already start with a lot of damage, actually, in young adulthood. And they tend to increase their damage load even further with age. Whereas the young ones, the predicted young ones, stay rather healthy. So just to stress this out, we can here really predict which neurons degenerate earlier than others at a young chronological time point. And now the interesting thing is, and this is only possible due to these aging clocks, is that we now can, of course, as I've shown you here, predict for every single cell type the exact age of these neurons. And now we can ask the questions whether there are any transcriptional patterns along the predicted aging trajectory. So are there genes or pathways that always go up with the predicted age or that always go down with the predicted age? And we did exactly this. We identified 4 different clusters. We have here the age bins. 1 is are the youngest neurons, 5 are the oldest neurons. And you see here cluster A, for example, are genes that tend to go up with age. Cluster B, they go down with the predicted age. C is a bit of an inverted U shape. In cluster D, these genes are not changing until the oldest neurons actually, where it's suddenly increasing. And what's interesting, of course, is that because we have gene expression data, we can also have a look at what pathways are involved here. And in cluster A, for example, so those that go up with age, we see a lot of translation-related pathway. We have rRNA processing, tRNA, we have the nucleolus and ribosome biogenesis, for example. And now I'm skipping a few details here, but the idea now is we know which pathways are going up with age, in this case, translation. If we now reduce translation, for example, this should counteract the aging phenotype. And we can now ask, are there any drugs, any treatments based on public available data that do the opposite of what we do see here? So with age, these pathways go up. Are there any drugs that do the opposite, that reduce this? And this is what we did. So we tried to find novel compounds that prevent neurodegeneration this way. And long story short, here, these are the top hits. So we have roughly 15 that are predicted to be anti-aging, and 15 again, roughly, that are predicted to be pro-aging. And directly something that was good for us is that we saw that most of the compounds that we predict to be anti-aging are actually known to be protective for neurons. Whereas for the pro-aging compounds, not so much was known. And now we took those compounds that were actually unknown and we checked some of them in the lab. And amazingly, we saw that the predictions held true, so that the predicted compounds significantly alleviate neurodegeneration in the living animal. So here just 2 examples for old neurons. We do see in the control cases a lot of damage. But now if we treat these living worms with our compounds, we do see more healthy neurons. So neurodegeneration is slowed down or stopped to some degree. And this would not have been possible without aging clocks. So with aging clocks, we were able to essentially predict the age of all these neurons, do these transcriptional patterns, and then use this information. So just to add to the previous conclusion, aging clocks can be used to find cell types that are susceptible to age-related degeneration. Here now we focused on neurons, but in theory, this could also be extended to other cell types. And the transcriptomic trajectories allow then the discovery science and the identification of novel, in this case, neuroprotective compounds. And this is only possible because we were able to use these aging clocks in this model organism. And with this, I'm at the end. Several of these stories I shared were done during my PhD, actually, in collaboration with Björn Schumacher. He was my supervisor. With this, I would be happy to take the questions. I'm looking forward to the discussion. Thank you.
Thank you so much, Doc. Can you stop sharing so that I can?
Sure.
Okay. Okay. Thank you so much for that wonderful lectures. Yeah, pure bioinformatics and that. We are now in for questions. So if you have any question, just raise your hand. If you have comments, concerns, you can raise your hand now so that we get into the whole conversation strongly.
There's a question
Okay,
in the...
Ikechukwu, you can ask your question first. Ikechukwu, can you ask your question? After that, I think the faculty is still here. The faculty can answer some of the questions here.
Okay, sir, can you hear me? Good evening.
Yes, hello.
Okay, thank you, Dr. David. My question is, I suppose that all you described today, it was a particular machine learning model that was developed to predict the ages. So I wanted to know, is it possible for Africans to develop their own aging clock, since this time around you will have to need a different data set? Like we adopt the model that have been developed to predict the age for non-Africans. Is it possible to just adopt the model, but this time around we change a different data set that will be able to predict for Africans? Thank you.
Yeah, this is a really important point. So what has been shown before is that several of these aging clocks that are already developed are indeed failing on other populations. So maybe to the first part, so these aging clocks are machine learning models. Many are completely doable with really simple models. You don't need like neuronal networks or something. Often a linear regression is already sufficient to get really good predictors. But I think what would be really needed is ideally some data set from an African population. So for example, just recently, I did not put this into this presentation due to time reasons, but we published an aging clock for a Southeast Asian population, specifically from a data set in Thailand. And we do see also some differences to other populations. It seems to work reasonably well also in European populations. But we, for example, could not test it in African populations. There was no data available. And I would expect that there are some differences. So one can try to adapt these models on African data directly, but I would expect that there are shifts in the data. So for example, that every African that you measure is maybe plus or minus 5 years or something like this, which of course is then not as useful. So ideally one would need data. And you can build these aging clocks on a variety of data. So you can have like gene expression or DNA methylation data, which is more expensive. But for example, the data that we used in the Southeast Asian population was based on clinical data. So your long-term sugar, HbA1c or cholesterol, and triglycerides and so on. So this is much more available and cheaper, of course. Probably this would be the first that I would try to adopt.
Okay, thank you.
It's all right. Before we proceed, Doc, I would like you to actually address that question. I think it's very important for the person that the question in chat was where she asked about simplifying the meaning of biological aging and all that thing. Yeah.
Yeah. Sorry if I went too fast or wasn't clear. So essentially, your chronological age is what is in your passport, right? It's based on your birth date, and you're maybe 40 years old. The biological age tells you, biologically speaking, how functional you are. So some people also call it functional age or physiological age. So for example, if I am 40, but I'm multimorbid, maybe I have type 2 diabetes, I have chronic kidney disease, and I'm really frail, then my biological age would be much higher because I'm, biologically speaking, maybe closer to a 60-year-old. So my body is 60, so it's aging faster than it should be. But then on the other hand, I might be 60, but I'm still running marathons, and actually I might be faster than the average 40-year-old. So biologically speaking, I would be much younger. And this is, of course, associated with frailty. So if I'm chronologically old, but biologically younger, then I'm not as frail. So I have more movement capability. I'm just fitter. I can do more sports and so on. And I usually then also get less disease. So this is the rough definition of biological age or functional age, physiological age, as some people call it. I hope that was a bit clearer.
Yeah, that works. Thank you so much. Jane, can you ask your question? Jane, if you're there.
Yeah, good evening. Thank you so much, Doc. So my question is, I want to know, can anti-aging clocks be used to measure the effectiveness of interventions like diet or exercise? I also want to know, are there any ethical concerns with using anti-aging clocks in clinical settings?
Good questions. So interventions, it's mixed. So some interventions seem to be predicted reasonably well. Other predictions, it's really dependent also on the aging clock. There was also a recent paper that compared different aging clocks and how they respond to different interventions. And it sometimes really depends on the aging clock. So not every aging clock is doing equally well. And some interventions are just not really appearing too well. So usually, because these aging clocks seem to measure inflammation, if you have something that reduces inflammation, it's often showing you a negative age acceleration, so a younger predicted age, for example. But at the moment, it's still difficult. The other question regarding ethics, you need, of course, the consent of all the individuals that this works. One problem is that often you have then genetic data, so DNA methylation, gene expression, you often have the sequencing, so the underlying genes attached to it as well, which you have to save really securely. You cannot just share them freely, of course. So it's really important to have a good security setup on the IT system. And then, at the moment at least, the results are not 100% clear, right? So you do not know really whether, if the intervention is predicted to make you 5 years younger, whether this is really beneficial then in the end. So at the moment, it's really difficult to make good suggestions. For example, coming back to inflammation, maybe shortly, briefly, some individuals are inflamed without an infection. So in these individuals, you might want to reduce inflammation, and then it's beneficial for them. But if you have an acute infection, maybe you need the inflammation. So you don't just artificially want to reduce it. Maybe it's important for them to have some inflammatory response. So if you then just see you reduce the inflammation and the aging clock looks better, but actually it's worse for the patient because it needed the inflammatory response. So it really depends on the underlying condition and what this aging clock measures. So at the moment, one has to be still really careful with how you use it, I would say, if that makes sense.
Yeah, that's powerful. Okay, Kifai, you can ask your question before I ask my.
Yeah, good evening. So I have 2 questions. Please, can anyone hear me?
Yes, yeah.
Okay. My question is a bit, I don't know how to put it, but my question is what frameworks, or approaches can be used to control lifestyle factors that impact the rate of biological aging? Like when you talked about the entities that are being used to gather the details or the data in building the aging clock. When it comes to someone's behavioral patterns, or should I say attitude, characters, what frameworks or approaches can we use to control these factors, in order for us to determine the rate or the impact of the biological clock?
Mm-hmm.
I don't know if you understand.
Yes, I think I understand you. It's a tricky question, I would say. So, I mean, currently there's limited data on the impact of, for example, lifestyle factors on the rate of biological aging. So you have some big cohort data like the UK Biobank, where you have a lot of data available and people do different kind of association studies to see what impacts the biological age. There are then, of course, some interesting insights, and often, of course, you do see that certain interventions, sports or sleeping and certain diets do reduce the predicted ages. But at the moment, the data is still not completely there yet, because usually you don't have, for example, longitudinal data. You don't have sufficiently diverse data sets. And often the effect sizes are also not super big. So it's difficult to really interpret them well. That being said, what one, of course, can try to do, and there are companies that are doing that at the moment, is that they measure your biological age, then they try some intervention, something not dangerous, of course, like certain diets, sports, and so on. And after half a year, they again measure your biological age. And depending then on the aging clock, you can then, of course, first of all, see whether you got older or younger. And also, depending on what these aging clock measure, you can see what exactly was impacted or not. So if you have a clinical aging clock, you might see that your HbA1c reduced, and this reduced also your biological age, for example. So at the moment, mostly is lifestyle. There are a few promising looking therapies that might have an impact on the biological age, but at the moment the data is not there yet to really claim something, I would say.
Yeah,
Okay.
a second question.
Just a follow-up question based on what you said. I want to know, because we as Africans, we are quite different from the Europeans, the Americans, and so. So what is the impact? What is the impact of fear in this biological aging? Looking to the fact that in Africa, you can, let me assume that you measure someone's biological aging. For example, I've encountered someone that said oh, don't check my BP. Checking my BP alone can kill me, you know, due to the fear, you know, all those kind of stuffs can actually trigger some kind of high blood pressure, which will lead to other sickness that will quicken the person's or increase or reduce the person's lifespan as well. So what is the impact of fear in this aging clock?
Yeah, that's so maybe I answer in two different steps. First of all, some variables like blood pressure, for example, are really variable. We in the past also tried to include blood pressure in an aging clock, but we decided to remove it because it's really easily highly variable. So if you have not slept as well, or if you have caffeine, or if you have fear, for example, you suddenly have a higher or lower blood pressure, which really then can mess up your aging clock predictions just on a technical level. So we remove these highly variable measurements on purpose. That being said, there are some association studies showing that perceived well-being does correlate with the biological age. So if you think of yourself as young and healthy, often these individuals also show a younger predicted age. So the causality is not completely clear, of course, but I do think that if you have a positive mindset, and there are several papers that show this, then usually these individuals also tend to live longer. So if you have a more negative view on the world and are fearful and anxiety, often this has a negative impact on your age. So usually you age then also faster. So I think that's a really important point that is not so well studied at the moment. There need to be more studies, I think.
Okay, thank you so much, Doc. Okay, please can ask your question, please.
All right. Good evening, sir.
Hello.
Hello, can you hear me?
Yes.
All right. Okay. I want to ask, my question is on fasting, like autophagy fasting. I want to know if it has any impact on slowing biological aging or fasting in general. How does it impact biological aging? Thank you.
Yeah, that's a really interesting field of study. It has been shown in not only many model organisms, but essentially across the board that fasting does seem to reduce your rate of aging. So this has really been shown from model organisms like mice, worms, monkeys, everywhere where you reduce calories, you do seem to increase your lifespan and also push back age-related diseases, for example. And here, I don't even think we need aging clocks because the data is pretty clear on that. In humans, of course, it's hard to do studies here, but also there have been studies looking at calorie restriction and then following them with aging clocks. And they do see that people that do fast have a slower aging rate. So over 2 years, for example, people that normally age, they just increased their biological age by 2 years, but those that fasted were almost flat. So they had a really slow increase in the biological age. So reducing calorie intake, I think is really valuable as long as you have, of course, your vitamins and so on an equal level. If it's just calories that get reduced, it's really powerful.
Okay, please, was I helpful? Charles Bassi, please ask.
Yes, sir.
Okay, Charles, please ask your question.
All right. Good evening, everyone. Can you hear me?
Yes.
Okay. Thank you very much. My question is on the aging clock. Like here in Africa, we have external stressors that are very common in African regions. You know, we have higher infectious diseases. We have like malaria and TB and all of that. Sometimes even different dietary patterns. So I want to ask, how does this affect the noise in an aging clock? And I also want to know, can these clocks distinguish between natural aging and cellular stress that are caused by the environment? Thank you.
Yeah, again, really important question. It's, I would say, usually also the data that is nowadays used, these are often cohorts that are supposed to be a general population. So there will be individuals that are sick and many that are healthy. And of course, the older the individuals are, the more likely they are to be sick with some kind of disease. And if you build a clock on this, then if you have sufficient data, it should be rather robust. If, of course, your training population is only sick, then this would bias the predictions of healthy individuals, for example. That would probably not be a good idea. You can try to build a clock only on healthy individuals and then see what effect it has on sick populations. But often it's okay to have a general population if you have sufficient data. If you have, of course, high prevalence of certain diseases, I would certainly make sure that the training data looks somewhat balanced so that you have a sufficient healthy population also in there, that the age range is also somewhat equal. But generally, I think that is doable if you have sufficient data for that. And if you have the information available, whether someone is sick with a certain disease, with an infection or whatever, you can then afterwards also calculate what impact this disease has on the predicted age. So specifically to look at whether a certain disease does increase it. That's certainly doable. I hope that answered the question.
Okay, thank you so much, Doc. Mine is more or less few questions to help carry some persons that are not from the science background along, and there are some persons, we have clinicians here, we have researchers, but we have people who are just interested in what we're doing. So that's why we get some certain questions. So of course, you know, many biomarkers used in aging clocks are also influenced by infection, which is one of the questions that they are asking, inflammations and metabolic disorders. So how do we distinguish biological aging and pathology-driven signals, especially in regions with high infectious disease prevalence, like Nigeria example, malaria?
Yeah, this is indeed a tricky question. So ideally, what you would like is a really thorough data set, so where you have the data that you want to train on, but then also a lot of meta information. So this could be a disease state, chronic diseases, and so on, the age, the gender, and all other kinds of meta information that you can think of, because then you can try to correct for certain influences. Maybe one example I can give is I mentioned this clinical aging clock that we recently built, and full disclaimer here, this was in collaboration with a company. So I consulted for them. I don't get money for the aging clock now, but I was paid to build this aging clock. And in their case, this data was from a hospital in Bangkok. So there, of course, we have many sick people. And what we decided in the end, and this worked really well for us, is to first really stringently go to the data and define what a healthy individual is. So we didn't want acute infections. We didn't want chronic infections or cancer and so on. So we excluded everything that could make a problem in the long run. So we focus on something as healthy as possible. Of course, people 60 years and older, there it's difficult to get a completely healthy population, but we try to be rather stringent. And then we built an age clock on this, a population, and this seems to work really well also then on diseases. So if we now use this clock that is trained on healthy populations, on, for example, individuals that have diabetes, we do see that the people with diabetes are 10 years older. And we even do see that people that in the future will get diabetes already have an increased age 5 years beforehand. So if I will get in 5 years diabetes, with this clock, I already have an increased age. So it could give an indication that maybe the doctor should look at something to check up. And one important thing there is also that we excluded, for example, C-reactive protein, CRP, which is usually responding to infections, for example. If you have an infection, this shoots up and it gets really high. This also goes up with aging, but with infections, it goes really high. And if we would have included that, then the biological age would be really sensitive to infections, for example, like influenza, COVID, and so on. But we excluded that, and we do see that if an individual has influenza or COVID, the age stays roughly the same. It doesn't change so much. But if it's a chronic infection, a chronic inflammation, in that case, it does show an increased age due to the other biomarkers that we used. So depending on what biomarkers you use and how the training data looks like, you can try to get a clean prediction. Another way to do it is that you need even more data, is you would need mortality data. So if you have time to death or mortality risk of the individuals, then you can directly try to predict mortality. And in that case, the hope is if you have an all-cause mortality predictor, that it learns different patterns. So if you train chronological age, which many people do because that is the data that we have, it's more difficult because then you don't know sometimes if it's an acute infection or not. If you train until mortality, often it's getting cleaner because there you just predict all-cause mortality risk. It depends a bit of what you want to predict in the end. But if you are worrying about some infections or some diseases, you can try to exclude them from your training and just train on the remaining data. Comes down to what data is available.
Yeah, that makes sense. I'll have like a 2-in-1 question I'm going to ask. You at the course of your presentation, your teaching, partly you talked about proteomics, but not well pronounced, So of course, we know that advanced aging clocks often rely on omics data, because it's more or less also a kind of omics technology for precisioning, right? So which may not be widely accessible in Africa. So what's simplified or cost-effective versions it can be developed that still provides some sort of meaningful insights for clinicians and public health system. So in summary, I would say, what is the minimum viable model for low resource settings?
Yeah, I would say the easiest way to go is probably with clinical biomarkers. So clinical blood draws, for example. There are, of course, also non-invasive biomarkers that are shown to correlate really well with frailty and diseases, like, for example, hand grip or walking speed and so on. So there are certain frailty tests where you don't need any blood drawn. These should also probably be generalized fairly well across populations. But if you want a bit more molecular understanding, the next step is, I think, these clinical blood draws, where you have your HbA1c, your cholesterol, your albumin, and so on. And these clinical aging clock that I mentioned before, there we used 6 biomarkers. and your gender. And we can predict the chronological age to a really high accuracy and certain disease onsets in the future. And because these are routinely measured biomarkers, this is really cheap in comparison to other aging clocks. So if you have a blood draw and you have a clinical checkup and you test your HbA1c and so on, it's just a few more biomarkers. In this case, it's 6 biomarkers that could be measured. It doesn't have to be these 6. These work well for us in this Southeast Asian population. But I think that you can get good indications, which are also more easy to interpret for clinicians because they work with your HbA1c and so anyways. So it's easier for them to understand actually what the predictions is saying. Maybe I will just share the publication in the chat that I just mentioned.
Okay. You can actually send because there are some persons that are struggling with network. Some persons are joining from YouTube. So I'll share it in a group. So maybe you send
I will send the link. Maybe you can send it then later.
Okay. Peace and Suliat, please copy the link and send to the group. Okay, my final question, Doug. What are the key steps? The key steps are needed to develop large-scale, ethically governed aging data sets in Africa, and how can emerging initiatives like Afrolongevity contribute to building a continental framework for longevity research and application through our R&D units?
Yeah, this is really something I think it would be interesting maybe to set up a meeting to just brainstorm this. I think it could be really good to schedule a meeting for this. I think certain points might be really difficult to implement, but I'm also, for example, in contact with a few people that try to standardize how clinics work on the biobank level, for example. Currently, the focus is for them in South America. But I think it would be really interesting to get you guys in touch to maybe build this network more worldwide, more globally, to then hopefully establish some kind of framework that we can utilize in the future, because it really comes down to a good system in the background and really standardized ways to measure and store things. If that is doable, I think it could be really worthwhile. So maybe it would be good to have a bigger discussion with this about this topic in the future.
Yeah.
Really important point.
You so much. Thank you so much, Dr. Mayer. We can thank you a lot for making out time to be here. You've been traveling recently, and you know, most of our faculties that make out time to be here, it shows their interest and passion in the longevity crusade. Aubrey would say that we're crusading biological aging, so we need the right information, especially to make sure that because for me the first step to bridge the longevity divide in every country or continent is to provide the right information, transfer of knowledge, the ability to communicate clearly what is needed to be done and how it should be done. And with me here, passionate Africans from different countries who make selfless sacrifices to be in class to learn and to find a way to translate what they've learned to a more narrowed community. So thank you so much for your time. We really appreciate you for being who you are. Thank you.
Yeah, thank you for the opportunity to talk to you, and I hope to continue this collaboration in the future. And if anyone wants to discuss something more, just drop me an email or message, and then we'll be happy to discuss this in more detail or answer your questions.
Bye for now. Thank you so much, guys. All the best.